Bismillahirrahmanirrahim. Assalamu alaikum students. Hope so you are all fine. This is your science class and we are doing chapter 1, food and digestion. The topic for today's discussion is sugars. Students, if we talk about the sources of sugar, then we can say that sources mean the way or the mode, the food from which we can obtain that thing, that particular thing. So we are talking about sources of sugar. So we can say that sugars are found naturally in fruits, sugarcane and milk. If we talk about the uses of sugar, then sugars are used to make chocolates, candies, cakes and soft drinks. So food or drinks made from refined sugar such as candies and soft drinks provide us with a lot of energy but little of other nutrients. So we can say that it is not advisable to take too much of them because they are not providing us with healthy nutrients. So if we take excess amount of these sugars, it can convert into body fats and cause obesity. And you know that due to obesity, we can face a number of health problems. Student, if we talk about the testing for glucose or testing for sugar, then we can check the presence of simple sugar glucose by using a solution, and the name of that solution is Benedict solution. Students, Benedict solution is light blue in color. So, when we add equal amount of Benedict solution and glucose solution and then we heat them, we will get orange red precipitate. In this picture, you can observe these orange red precipitate. It means we have added blue Benedict and sugar, then we heat them and it is converted into orange red precipitate, which is showing that glucose is present. If there is no glucose present, then blue color will remain in blue form. The next topic is fats. If we talk about the composition of fats, then we can say that like carbohydrates, fats are compounds made up of carbon, hydrogen and oxygen. But there is a little difference between the composition of carbohydrates and fats, that fats have less oxygen in proportion to hydrogen. So fats give more than twice the energy. So they are the source of energy for us and they give us twice the energy as the same amount of carbohydrates. So if we are taking 1 gram of carbohydrate and 1 gram of fat, then we can get, for example, if we are getting 5 kilojoule energy from 1 gram of carbohydrate, then we will get 10 kilojoule of energy from 1 gram of fat. So we can say that fats give twice energy as the same amount of carbohydrates. So we can say them, they are reserve supply of energy. So fats serve as reserve supply of energy when the food consumption is low and help to keep us warm. Fat will be used to keep us warm and will provide us with energy. So students, fat also cause different health problems because too much fat in our bodies can lead to health problems like heart diseases. So if we talk about the sources of fats, then we can say that we can get fats from animal and plants. So due to on the basis of sources of getting fats, fats are divided into two types: plant fats and animal fats. So students, fats from animals can be found in butter, lard, meat, cheese, and milk, while fats from plants can be found in margarine, nuts, and cooking oils such as peanut oil, palm oil, or olive oil. Plant oils and oils from fatty fish such as salmon and mackerel provide us with healthy fats, while animal fats such as lard and butter have high levels of unhealthy fats and should be consumed sparingly. So we can say that plant fats are healthy and animal fats are not so healthy. Students, if we talk about that how we can test fats, then the presence of fats in food can be tested by using ethanol. So in this test tube you can see ethanol is added to a drop of oil. This is a drop of oil and mixed well. If you get a clear solution like this is formed when you will mix it, and then you will add some water to this clear mixture and a white emulsion is formed. Basically this white emulsion is fat which is dispersed in water, and ethanol is a kind of alcohol. So when we will add a drop of oil with alcohol, then we will mix it, we will get a clear solution. Then we will add water in it, and due to addition of water we will get this white emulsion, and this white emulsion is fat which is dispersed in water. So in this way we can test fats. If there is no fats, then we cannot get this white emulsion. Students, the next topic is proteins. So if we talk about the composition of proteins, then we can say proteins consist of long chains of amino acids which are made up of the elements, and those elements are carbon, hydrogen, oxygen, and nitrogen. So here you can say carbohydrates and fats have carbon, hydrogen, and oxygen, while proteins have nitrogen in addition with these carbon, hydrogen, and oxygen. So there are about 20 different types of amino acids which link up and combine in various ways to form millions of different types of proteins. So there are total 20 amino acids, and they will join in different manners, different ways, and they will form millions of proteins. So if we talk about the function of proteins, then like carbohydrates and fats, proteins can supply us with energy. However, their main function is to build body cells like tissues, enzymes, and hormones. And they are also essential for growth and repair of worn out parts of our bodies. Like muscles, cartilage, and tendons in our bodies are made up of proteins. Our nails, our hair, these are also made up of proteins. So if we talk about deficiency of proteins, then students, deficiency in proteins results in a disease, and that disease is called kwashiorkor. Basically, kwashiorkor is a disease which is caused by the deficiency of proteins, and the characteristics of this disease is loss of body fat, distended belly, and wasting of muscles. So students, if we talk about the sources of proteins, then we can say we can obtain proteins from food which are rich in proteins such as meat, eggs, fish, milk, cheese, beans, nuts, and peas. So students, if we try to test a food that either it has protein or not, we can test proteins by using a solution, and the name of that solution is biuret solution. So presence of soluble proteins in food can be tested by using biuret solution, and biuret solution is a mixture of sodium hydroxide solution and small amount of copper sulfate solution. So student, we can make biuret solution by adding sodium hydroxide solution with small amount of copper sulfate solution. So student, the color of biuret solution is violet. Here you can see the color of biuret solution is violet, and if we add a protein, that this violet color will be changed into blue. So when the blue biuret solution is mixed with a soluble protein, so if the solution is turned into violet color from blue, then we can say that protein is present. So students, I am explaining it again that the color of biuret solution is blue, and this blue color will turn into violet color if protein is present. But if there is no protein, then the violet color will remain violet and it will not be changed into, the blue color will remain blue and it will not change into violet color. But if protein is present, the blue color will be changed into violet color. So students, this was all about sugars, testing for sugars, glucose, fats, testing for fats, proteins, and testing for proteins. I hope you understand this lecture very well, because we have discussed about proteins, fats, and sugars, and we know the difference between sugars, fats, and proteins. So sugar is made up of oxygen, hydrogen, and carbon. Fats are also made up of oxygen, hydrogen, and carbon. But in proteins, nitrogen is present in addition. So we have discussed the different sources of sugar, that how we can get them and how we can test them, that we can test sugars by using Benedict solution. And fats are also like carbohydrates, but they have less oxygen in proportion to hydrogen. And we can test fats by using ethanol, which is a kind of alcohol. And we can say that proteins are different due to addition of nitrogen, because carbohydrates and fats doesn't have nitrogen in them. And we can test proteins by using biuret solution. And the color of blue biuret will turn into violet color if protein is present. Students, if we discuss the test of sugars, fats, and proteins, and if we compare them, then we can say that testing for sugar is done by using Benedict solution, and this Benedict solution is light blue in color. And when we mix equal amount of Benedict solution and glucose solution, then it will turn into orange red precipitate. And these orange red precipitate shows that glucose or sugar is present. But if we talk about testing for fats, then we use ethanol for fats, which is a kind of alcohol, and the formula of ethanol is C2H5OH. So we will add drop of oil and we will get a clear mixture, then we will add water and we will get a white emulsion. And this white emulsion is fat dispersed in water. So through this method we can test fats, but if there is no fats, there will be no white emulsion. And the last test was test for proteins, which will be done by a blue solution known as biuret solution, and we will add protein in it. If the adding material is protein, blue color of biuret solution will turn into violet color, but if there is no protein, then blue color of this biuret solution will remain blue. So testing of glucose done by Benedict solution, testing of fats done by ethanol, and testing of proteins done by biuret solution. I hope students you understand this lecture. If you find any difficulty in this lecture, you can ask me. Thank you so much. Allah Hafiz.